Hello, everyone, and welcome to another Canna Talk session. Once again, I'd like to thank Canna for organizing this event and being leaders in educating, supporting, and developing our industry. Today, we continue the conversation about testing discrepancies and how it affects our industry. A little bit of housekeeping before we get going. This is an interactive session, so we encourage participants to ask questions and we'll take them as the panel is ongoing. Please note that everybody's muted except the panelists here. If you're watching on the Canna website, you should see a Q&A box just below your screen. You can put in your question there. Or if you're watching in full screen mode and don't want to scroll around, you can use your mobile device, scan the QR code on the right-hand side, and ask your questions there. So part one of this discussion was actually held in Winnipeg during the Strathcan Growing Relations Conference, and we discussed who and what drives this THC race. And surprisingly, responsibility and blame was not clear and not all one-sided, which was really interesting. But they all agreed that integrity should come from all sides, that there is a lack of consumer education, and finally that the regulators also need education and guidance to increase some of our consistencies at all stages of production, processing, sales, and distribution. So for part two of this discussion, we brought a new set of lineup of panelists to dive in a little deeper into this THC race and how it affects our domestic and potentially our international markets. So on our panel today, we have Jonathan Maupetit. He's the Senior Director of Plant Sciences, Master Grower at Organigram, and they are focused on producing high-quality indoor-grown cannabis for the adult recreational consumers in Canada, as well as developing international business partnerships to extend the company's global footprint. Next, we have Dave Clermont, founder and VP of Sales and Genetics at Tricalix Nursery. Tricalix is an indoor cannabis nursery who is dedicated to cultivating high-quality genetics for the benefit of licensed cannabis producers from Quebec and Canada. And lastly, we have Thomas Frawley. He's the president and founder at Vivariant Laboratories. Vivariant is an ISO and GMP accredited analytical chemistry and microbiology testing lab in Mississauga, Ontario. The company services clients in the pharmaceutical, cannabis, natural health, dietary supplements, and cosmetics sections. So for more details on our panelists, please visit the Canna Talk invite for their full bios or click on the panelists section on the webpage below. We'll get right into it. I did ask some similar questions in Winnipeg, but I think it's important that we reset some of these baselines and continue the conversation ongoing. So I will refer a little bit to our previous conversation to keep the flow going. So I'll start right away with Thomas. Thomas, can you explain what we mean by this THC inflation, and what are some of the drivers behind it? And I just would like to mention that we're not here to throw anybody under the bus or name names necessarily. We just want to bring light to the issue. So yeah, what is THC inflation, and what are these drivers? Like, who drives this?
The term THC inflation refers to sort of this broader trend of THC labels on cannabis products, particularly dried cannabis flower, increasing over time in markets. The challenge with these values is that they have commercial implications in the supply chain. So you see generally consumers drawn towards purchasing decisions based on THC values that are on the product. This is exacerbated in a market like Canada, where consumers aren't able to actually open the container and look at or smell the contents. So it's one of the few metrics they have to make a decision, buying decision based off of when they're in the store. Maybe perhaps terpene values as well too at best. And then conversely, because at the retail level, higher THC labeled flower sells more, it means that the wholesalers and distributors are more likely to make bulk purchases based on that value as well too, which means that the producers are under pressure to label their product with essentially a higher and higher THC values. So because there's this what I'd call like upwards commercial pressure on these values, it makes them susceptible to being compromised and losing integrity along the supply chain. A testing lab will win more business if they give their flower clients higher THC values compared to a competitor lab that doesn't. And likewise, producers will be able to sell more product the higher lab test value they're able to obtain on the product that they sell. So, like I said before, this kind of creates a positive feedback loop where now all of a sudden these sort of gradually more inflated values are being assigned. Now producers have to compete against their competitors who are using these higher values, and it creates this feedback loop where the numbers start to rise up over time. I'd say this trend was like part of it that's kind of frustrating is, we legalized cannabis, in 2018, but this trend had been observed in, several US jurisdictions that legalized at the state level prior to Canada. So it's a little bit frustrating that this was like not anticipated that this would happen because it already happened even before we legalized. So it's a bit disappointing that our regulators didn't account for this from the start. As a consequence of THC inflation, we begin to see products labeled with THC values that like may or may not be realistic anymore from like a biological perspective. There were numerous instances over the past year in Canada alone where journalists pulled like SKUs from just say OCS or other regional markets that were claiming these kind of like 40% total potential THC values. And there was a little bit of a journalism cycle where they were sending those in to get retested. We participated in a little bit of that. Some of the values that even we got back in the articles we participated in were substantially lower. I think we had a journalist send us a sample of a product from the market that was labeled at 39% THC, and that we got like 23 when we repeated our analysis here. So that's sort of the overview of what the term THC inflation means and the implication in the market that we all sort of participate in.
Thank you for that. That's quite a bit detailed, and I really like your point of saying, we've seen that trend happening in the U.S. in more mature markets, right? These smaller markets mature much faster than ours, but the trends are there, and I think you're bang on that, Thomas. Looking at it not just from a marketing and branding and sales perspective, but also on the regulatory side of things and trying to anticipate, like you're saying. That's a very good catch on that. Jonathan, I know that Organigram, have been around since the first round of licensing, and so within 10 years, you've done a tremendous amount of research with genetics, bringing new genetics on. Can you highlight some of the findings regarding THC that you've seen? Because I know you have a lot of data. Don't share any proprietary information, but we'd love to hear your thoughts on, some of the trends you've seen or what is possible, what is not possible, I guess.
Yeah. I guess maybe just to give an overview of how we do our testing when we do our genetic screening, we're testing multiple times throughout the cycle, and we take triplicates to make sure that what we're is reality. We last year alone we screened 1,200 genetics, and I think we hit 30%. That's anhydrous, so dried to 0% because we're taking fresh flower and we're running it through. We hit that maybe a handful of times, 5 times or so. So it's really a minority of genetics that are reaching that high, above that 30% threshold. And again, if I'm talking about anhydrous, you got to take 10% off that number. So if something that's coming in at 30%, you're really getting 27% release testing. And then it's representation of your lot as well, right? So how are you testing your entire lot to make sure that what you're putting out to market is really reflective of what you're seeing? That's another thing. So yeah, with all of the genetic screenings that we've done, and we regularly also bring in genetics from other people that we're testing, just to make sure that we're getting the same numbers or similar numbers, testing them in our environment and seeing. But that 30% threshold is, unicorns and rare ones. So it is very frustrating, and it's also very surprising to see all of a sudden the market shift from one year where everything is at the 25-26% range, and then the following year you're seeing tons of high 30s out in the market. It's quite surprising.
Yeah, it is quite surprising because I mean the trend was climbing over time. Um, I think we're getting some feedback there a little bit. Um, and but it was a slow climb in the last 5 years, and then you're right, in the last year it really jumped up, and that's something that we discussed in Winnipeg as well, and really what's the driver there of that, and essentially it's consumer demand that was driving all this. But then when we dove into that a little bit more, uh, it's actually there's a gap in education or knowledge from the consumers, and they don't actually understand what they're buying. So us always saying that high THC is a quality marker, I don't believe myself it should be a quality marker. It should just be a facet of that product. We don't qualify alcohol percentage as a quality. We qualify the rest of the product as a quality. Anyways, that's my little tidbit on that. So thank you, Jonathan, on that. So Dave, coming from a nursery, and all the amount of genetics development you've done, similar question to you, like what kind of trends have you seen, and what's the unicorns? And I'd like to add a little bit to you is, what are we leaving on the table that we're not actually bringing to market that should be brought to market? Like, is there something else that we should bring to market?
A world of different terpenes that we're missing out on. Definitely stuff that when we started to work on our genetic bank to create the nursery project, my goal was to bring in as many of these genetics possible to have pretty much like a cook, as many spices and ingredients possible to play with those, and to be able to offer different genetics that would be from either the older school genetics, from the new school, the new stuff that you're seeing coming out of California. Well, and then what happened is that everything that we brought in that we thought and felt that would be bangers on the market immediately fell into these constantly higher THC cracks where they couldn't have any shelf life or any potential commercialization because they didn't hit those numbers. And when I started, it was talks about, oh, it has to be 20% and over, and then 23, and then 25, and then I've seen ridiculous things as high as people seeking out lots of batches of dried stuff that needed to be up in the upwards of 34% or higher. It's just I don't. I'll exactly reflect what my previous speaker just said. Our
Mm.
testing shows definitely numbers that aren't higher. And if we hit a 28%, 29, 30%, that's a true banger, and it's a rare thing. It's a unicorn. We have definitely a ton of genetics that have been left over because of that, but they have intense and really nice terpene profile that are just getting pushed aside because what you're getting into the market right now from those high THC strains, if you look at the backgrounds of a lot of these strains, there's a specific strain that that are pushing these numbers higher, and then you'll get the same terpene profile coming over and over and over again because you're breeding with the same stuff to push those numbers higher. So
Mm-hmm.
eventually, it'll become kind of a stale, or it just cancels out other older strains that we love the profiles of, and it but this trend has to eventually stop because it's gonna kill the cannabis diversity for sure.
Everything's going to
Uh,
taste like GMO.
yeah, and I couldn't
Spot
agree
on.
more. I couldn't agree more. I mean, what's this racist has to end at some point. We are also putting a precedence internationally on this as well. Internationally, people are seeing what is for sale in Canada. The problem is when we get it tested back in their country, it doesn't match, right? So are we setting ourselves up for failure in that sense? That's just a generalized question for the international market. It's all fine and dandy that it works right now in the recreational. I think we need to educate our consumers a little bit more on what is quality, and I don't know what it is, but yeah, that's my 2 cents on that. And we have to be very, very careful on the international market. Yeah. So, Dave, you mentioned that, both of you actually, Jonathan and Dave, mentioned the unicorns, and I believe how much genetic development you guys have done. I follow both of you guys. Jonathan, in Winnipeg, they mentioned, I thought this was very interesting. They mentioned that growers, methodologies, facilities are getting better. We're learning, we're getting more refined. And that is actually a big factor in this increase of THC. Do you believe this is as grand effect on our THC race? And B, I'm asking you specifically because I know Organigram is very, very refined in their process, and you follow all that. So, yeah, what's your comment on that? Like, the growers are getting better, the industry's getting better. Are we going to have a never-ending climb?
Yeah, that's a fantastic question. Yes, totally agree. Growers are getting better. Growers, as you move forward and you dial in that cultivar, it's going to get better over time. I remember when I joined in 2019, 17%, we were happy. We're tapping ourselves on the back because we're hitting 17, sometimes 18. We kept refining it. Then we hit 20 as the market evolved. We hit 25 as the market continued to evolve because every year we kept pushing it. But that's the max that we've ever hit with that genetics. And that's just the genetic predisposition of that plant. So what we've done is we've bred in some other things. We talked about GMO or high potency cultivars to bring up the potency. I think you can definitely refine your process. You can work, retrofitted with LED. We've worked on our environments. We've done lots of things that have improved the potency. But again, every plant has a predisposition to reach that potency. And when we start talking about these 36s plus a 5% terpenes, like, that's a lot for a plant. That's just on a single compound. There's a bunch of other things in the plant, a lot of energy that's diverted to many different things. It just doesn't add up. So I think spot on. The refinement is going to be based on the yield that we're starting to see and the cost per gram that's going down. A lot more quality that's out in the market. And yes, we're going to see a little climb to potency, but I stand that 36%. I don't think that's actually feasible for a plant. Not 36% when it's dried to a 12% moisture content.
No, absolutely. I absolutely agree that the technology that we have now are better than what we had in the past. What we're seeing with some of our partners definitely is when they get their first batches in, because this is when everything bad happens with your HVAC, with your lighting, with everything. This is when usually they'll get the lowest percentage. So as they fine tune their environment and their techniques, the numbers goes higher. So this is obviously a good point where you're right. The growers have gotten much better than in the past. We've definitely climbed up those THC levels because of that. But yeah, there's a certain limit where we can go, I think, and where the plant can't give more than a certain percentage. And I don't know what's that number, but I think we've peaked pretty much what we can get out of those best strains, those highest THCs that we're seeing. Maybe there's going to be some improvement that are going to push us higher a little bit more, but I mean, where is that cap? What is the absolute maximum that you can get into this little bead of resin?
Very well said,
and how
Dave.
about testing methodologies?
That's
Thomas,
that's exactly my next question to Thomas. But good segue, because actually there's a couple of questions coming from the audience here. Thomas, why and how are we able to get different lab results from one lab to another? Is it the variation from the lab side, or is it other factors like sampling and all that stuff that comes into play? Like, without being the whistleblower, who's the culprit? Like, why do we have such discrepancies here? Because we've heard now on a couple of occasions that they've said they've cross tested, and they said the same thing in Winnipeg, and results were different.
So I think to start with, it's important to define like difference in test results between labs, because there's I would call like normal differences and then maybe not so normal differences, right? Like you take any industry, whether it's clinical testing, environmental testing, and you send the same sample to 10 different labs that do everything right, you're always going to receive slightly different values. And that's just normal. That's part of the chaos of our universe, statistical uncertainty associated with the method. Like if you have a sample of soil that's perfectly homogenized at 10 parts per million mercury, you send that to 100 different environmental labs, you're going to get 9.8, 9, 10.1, 10.2. That's all totally normal and just part of regular statistics for doing analysis. So when we talk about variation in between laboratories, that kind of variation is really not an issue at all. That's totally normal. It would actually be very concerning if every lab got 10.00, because that would be actually more of an impossibility. So this kind of normal variation can come from a variety of different factors. So small differences in methodologies, uncertainty associated with your method, calibration of your pipettes. This is all very like normal. The sample itself can also be, to a certain degree, there's heterogeneity inside of the cannabis plant in the same lot as well too. The buds from the top, middle, and bottom, or different areas of a grow, can all have varying amounts of cannabinoids, which can influence some of that results. If you are interested in sort of comparing results in between labs, I'd suggest sending in milled flower, because that's the best way of comparing results between different laboratories, because you eliminate the variation seen in the buds themselves. I'm also going to point out as well too that like on the lab side, it's harder to test things that are trace levels. So trace levels of pesticides, trace levels of mercury, that's where you're going to see more wild uncertainties. Cannabinoids are very abundant in the plant. It's not a hard test. When you're testing something that's a 20% concentration that's a dream for a chemist, right? Because it's present at such high concentration, you just have to make sure you're grinding it and doing your dilutions properly. So again, I want to give context to what is normal laboratory variation first, because yes, that exists. That exists in every single industry where there's testing involved, and it's normal. Then there's a difference between, I would say, like unscrupulous deviations or changes or differences in between labs. So unscrupulous practices can come from both the client side and the lab side. So for example, if a client wanted to bias their results, they could dry the sample prior to analysis and prior to sending it in. We hear of situations where potentially people are spiking in keef for THCA diamonds in order to boost the value. Or you can even bias the way that you pull your samples for potency. Like if you only select the most crystalline bud tip heads and send that in and say, this is my potency sample, that's going to influence the result that comes out. From the lab side, for unscrupulous practice, which I will be very explicit in labeling as fraud, there are any number of things that could be done that could boost those values. You could underreport your sample weight that you used in the workflow, like saying I used a 200 milligram subsample, but actually using 250 or something like that. That'll boost the value that comes out. You could use more dilute calibration standards than what were claimed in the method, drying the flower at the lab side prior to analysis. Again, it's a pickle to untangle. And again, I don't think any one side is doing this, right? Again, THC inflation is a term that is describes a general trend in the industry. It's no one party doing this due to those financial pressures that are on the number. It comes from both sides. One thing I always like to point out, just as a thought exercise, is that typically on the client side, it usually only takes one person to compromise the sample in a way that could boost those results. From the lab side, it does take a little bit more corroboration between multiple levels. You would need to have management telling the bench level analyst, we need to do this in order to boost the results. QA would need to know not to flag these things for review. So I would say that for labs that are bad actors, the corruption needs to be a little bit more systemic and organized, which I would argue is worse. But certainly there are actors on both sides of the coin that can contribute towards why we would see a compromised or inflated number at one lab versus another. So one of the things we quite often hear is that, oh, well, it could be the client, it could be the lab. We'll never know. I guess we can never solve it. And this is why I would say that this is where we need third-party intervention from regulatory agencies actually taking a more proactive role in this situation. Because if not, we're just going to be caught in a constant loop where the lab's blame the client's blame the lab, et cetera, et cetera. So that's a little bit about how you can see these large swings in results. It's just important to untangle what is normal, regular, like expected deviation and what is something that's a little bit more a result of corruption or unethical practices.
Thank you. That does paint a very nice picture. It's very similar to what was discussed in Winnipeg, and I'm glad that we're having a similar conversation because, to be honest with you, I thought this was a whole lab thing, right? And it's clearly not a lab thing. It's not a game like Thomas just mentioned. It's an amalgamation of a little bit of everything: demand, supply. But just quickly, a quick follow-up, Thomas. In the last session, they brought up some testing regulation that they're not properly regulated for the methodologies developed by the labs. Do you know if there's anything in the works to start kind of bringing consistency on that aspect, or is this even an issue?
I don't think this whole, dream of a standardized reference method that everyone talks about is going to fix the problem. Because to be quite honest with you, even if we all USP, the United States Pharmacopoeia, European Pharmacopoeia, German Monograph have all started to come out with their own reference methods, which we'll see implemented more and more. Like I said, in a normal, healthy system where no one's a bad actor, even with those methods, you're going to see a small amount of variation. If you have a system where either side of the coin, the client or the lab, is already being a bad actor, then no amount of method standardization is going to fix that, right? So I do believe it's important to have reference methods. It helps us. It's great to have those. I don't believe it's going to be a magic fix to dealing with this issue.
Okay, interesting. And it'd be, and I assume that's why the regulators haven't really stepped in full force because of that. So Dave,
I'll just point out on the method side of
go
things,
ahead.
if you'll let me interrupt you a little bit more, is that you have industry organizations like C45 that have been bringing this topic to Health Canada's attention literally for years now at their in-person meeting. And you see from Health Canada this kind of attitude where it's like, just because you mentioned methods and method standardization, and even though I just said I don't believe it's going to fix anything, I point this case out just because I find it fascinating and infuriating. Which is that they say, oh, it's not our, one of the feedbacks they've been giving is we're not sure if it's our responsibility to regulate this. We license labs to handle cannabis. We don't accredit them for their methods. But I would point out that Health Canada has very explicitly published guidelines on requirements for testing for pesticides. There's 2 maintained federal documents on how that method needs to be executed, what the LOQs are, how it's to be validated, what needs to show up on the test report. And then for THC testing in hemp, there's a full-on analytical method, sampling strategies, all these very clear delineated protocols online to make sure that we, God forbid, don't have hemp with above 3% THC inside of it. And that's all very regulated by Health Canada, including actual reference methods that they've put out there. But all of a sudden, now it comes to testing THC in dried cannabis flower for consumer and medical patients. And the result is kind of like, we don't know if it's our job to standardize methods or care here. It just seems a little bit counterintuitive to me. You're saying you regulate and you enforce and you keep track of testing for THC in hemp, but not THC in dried cannabis flower. It doesn't really make a lot of sense to me.
Thank you. So Dave, I want to circle back on some of your business and how this is affecting the THC race is affecting your business. You mentioned your old genetics. You brought in a whole bunch. Are you parking those for now and then just looking for new ones, or what are you what's your next steps in this genetic thing? Because we're not done, and I'd love.
So obviously, right now, everybody wants to keep their business alive, and what's gonna make us sell clones is definitely a genetic that hits at least a certain amount of THC. So whenever we're pheno hunting, the decisions of which seeds and which genetics are being brought to the table for the next pheno hunts are all based on those THC levels. Obviously, there's also the terpene profiles that we're trying to diversify as much as possible, but it's getting to a point where it's not as fun as it used to be when the only thing you wanted to have is just good weed, just good looking weed, good smoking weed. And this is obviously, as Jonathan was saying, we're throwing away just so many cuts. It's just so sad because many of those, and this is where it's funny, in smoke tests, when you're doing blind smoke tests, the ones that people will point to that are the winners and that everybody says they're the biggest hitters are not going to be the highest THC. And a lot of the times when we get those cuts tested, they're around 20, 22. And the other cuts that are higher, they don't give you a higher buzz. They don't give you anything. It's just sad that this particular cut that had everything, and this is where it's difficult. You have to check all the boxes when you want to come up with a new genetic. It has to be a very easy strain to grow. It has to get volume. It needs to be beautiful, super terpy, disease resistant. It needs to have all these boxes checked, and then it needs to be super high THC. So
Yeah.
this is the last straw when a lot of the cuts don't make it. They checked all the other boxes. They don't check this one. But in the smoke test, they do check it. So if you do a blind test with everything that's on the market right now with those high THC, and you put lower THCs side by side that have different terpene profiles, I'll be really interested to see which ones would come up into these. And it's the same with the cannabis cups. When have they brought super high THC cuts to winning a prize? When it's usually, if you get those cuts tested, if you get those winning cuts tested, I'm pretty sure that you'll get different levels and not super high THCs. It's just because they have such a great profile of all the cannabinoids and all the terpenes. So definitely for us, it is a bit of a challenge for finding these genetics. It's narrowing down the choices that we have to do. I wish we could. And to answer your question, what am I doing with all these cuts that I find that check all the other boxes but not the THC?
Yeah.
Because I'm such a passionate grower, I'll often save those for either trying to pimp up a higher THC strain that will be lacking terps, or to get a variation in it. But obviously, it is a very tedious task to also keep that many cuts. So there's just a certain limit of how many different genetics I can save and keep for, I don't know how long until they can be used on the market. But yeah, definitely it is difficult. And because, like I was saying earlier, that market has shifted so much that it's nothing from day one that we started. It has gotten to a point where everybody who calls us for a new genetic wants those 25 plus and 28 plus and those crazy numbers that we can't even reach.
That's so we're almost coming back to, again, the objective has not changed. Quality production has not changed. It's the message behind it and what drives these consumers, because really that's what drives the whole industry. So Dave, just quickly, from a nursery perspective and all the development that you do in the genetics and your knowledge, this is a question from the audience, actually. How do we shift that narrative to the consumers? What can we do to keep going on educating this? Because I 100% agree with what you said. There's some excellent genetics out there that have been left. I've done blind tests myself, and I wasn't surprised by the results. Let's just say that it was the same thing as what you just said. And good quality is good quality. So how do we shift this education or this narrative to the consumers? Do we do
Well,
it
because
ourselves, or do we?
it's a batch by batch result, so we know that all batches fluctuate. It depends on what's how your environment was and how you've cured, managed, and everything. You know, the whole process makes your batch a little bit different each time. Uh, we know that it is so an average because there's higher, lower buds potency that it's all mixed up in your little jar. So this is why when you go back to a lab, and if it's written 27% and you brought it back to any lab, you might get 23, 25, or it's going to vary. Health Canada seems to have kind of a good buffer in terms of what they accept for that, but I guess because in terms of consumer uh, expectancy, I don't know. It's been brought up in other discussions before, but could it be just a low, mid, or high THC label, and get rid of that percentage that obviously it's is it like with alcohol if it's a 40% substance, you know what to expect from that, but with THC and all the other cannabinoids involved, you don't really know what to expect from that. So it's kind of obviously there's a bit of difference there, but it might be a part of the solution, might be uh, I don't know. It's driving a lot of the growers obviously to our lab consultants are saying is it's driving them to push those numbers just because it's going to make it or break it for them. If they don't sell a specific batch, this is those income from them for them is going to be at some point a very difficult challenge if they can't sell those batches. So this is why some people I think are getting creative with that because of the competition of all these higher THC products that aren't really as high as what the label says.
Yeah, Jonathan, just to kind of circle back the same question to you about the education, how do we do this? Because I know that hopefully I don't get in trouble for this. Organigram and yourself been vocal on social media about testing and some of the what we talked about, the genetics testing that you guys have done, some of those numbers. So where else do we need to go from there at this point? Because I think you guys are already doing something. Are you guys going to try to push this more, this education, this knowledge out there? I know that Organigram's always at the forefront of some of these things.
Yeah, I think as a leader in the industry, we find that this is extremely important, and we wanna try to push that narrative and move that. I think everybody that's in the industry has a role to play, and frankly, if you're not pollution, you kind of become part of the problem in many ways, right? Um, to me, it's education. Today we had the opportunity to the Cannabis New Brunswick bud tenders, and this is a regular topic. It's everybody talks about it when you go into a cannabis store, when you're talking with your colleagues from different licensed producers. Everybody mentions it, and it seems to be almost accepted in a way where some people gravitate to specific areas, or the sampling methodologies are discussed and things like that. I think we've seen in the States, there are some methodologies and there are ways that have been regulated. We've heard of areas and states that when you have a high THC COA, you need to have a second lab to validate it. There's been some discussions of having potentially this come in to pick up and go get the samples, identify where the samples should be taken in the room to really get a representative sampling of the entire lot, because I think that's one of the big things. You need a representative sample of your lot to make sure that the consumer is really getting what they're buying. I think right now the consumers are the biggest losers. They're the ones that are getting flawed because they're going in thinking that they're getting these high concentrations and they're buying based on that. And it's a hard topic to change and to educate them. But I think if we had that standardized methodology or the sampling part, at least everything would be at that level. I think we can try. I think we've all been talking about terpenes for the last 3 years, thinking one day terpenes are going to be the thing, and they seem to be, slowly coming to the table and to the topic. But THC just remains the top priority at the moment. And honestly, I don't have the answer of how we're going to fix it, other than continuing to discuss it and continuing to educate people so that when they are going in the stores, we talk about that aroma profile. Dave mentioned earlier that we're going to a spot where everything's going to be bland, everything's going to taste the same. And we've seen time and time again that the best aroma profiles and the best tasting cannabis is actually correlates majority of the time to the lowest potency, because you have just much more aromatic profiles that are in there. So we need to educate the consumers, and we need, sometimes to challenge people when they're saying, "Oh, look at this, it's X amount." Well, yeah, but what else is there in there? What are you really looking for? I think it'll come with time, or at least I hope it will come with time. But I don't think we have the answer at the moment.
Yeah, no, very good. We in Winnipeg we mentioned briefly that instead of the government paying money for those commercials on TV of having the guy come out of the party and he's talking to a cartoon joint and a cartoon beer and he's arguing about driving. Maybe they should put money into more education of what is this product instead of making cartoon jokes for adults. Anyways, that's just a little sidebar from me. So Thomas, we mentioned that is there who's responsible, who's not responsible. We're not looking to pinpoint. However, the labs are the holder of information. I know this has been done in the past, in the last 10 years in the industry, that some labs have blown the whistle on certain facilities for whatever. Do you think that should be something that a lab should have as integrity? Should they be whistleblowers on this if they see, the bad representative sample or some spiking of the buds? What are your thoughts on that?
Every time before you're about to ask me a question, you always say without naming names or pointing fingers, because in our pre-boarding call, I said I really wanted to, and you explicitly told me I wasn't allowed to on this call. So you're asking me what I think the role of for a lab in reporting a suspicious sample is. I don't think it's practical, because there's two reasons. One is we just receive the material and test it as it is. I don't always know what's batch released for my customers, what's an R&D project that they've done, right? Maybe someone took some flour, dried it in an oven just because they were curious to see what would happen and wanted to see how the lab results test. They're never intending to put that on a C of A. It's not the lab's responsibility to determine that or flag them to the regulatory authorities. And I'd say that coming back to it, we face the same sort of, what's the word I'm looking for? Commercial pressure, where a lab that's already going to deliver, let's just say, inflated results in order to gain more money, sure isn't going to report their client to the regulatory authorities, out of any sense of obligation to do well, because then they're going to lose that client's revenue stream. So again, I like to look at it from a human system, and humans are susceptible to corruption. It is a fact. And so I don't think what you're asking me is a feasible mechanism to regulate the industry.
Okay, thank you. And just FYI, there was a quick comment on the Q&A section. Somebody really liked what Jonathan said about the second lab validation. So again, bringing these topics to light and talking about them, maybe we will come with some risk mitigation methods. And William also put in, what if Health Canada removes the THC value on the labels to replace it with sub-matrix like low, medium, high, just what Dave said. So is that a solution? I don't know. In my opinion, I could throw this to Thomas, but in my opinion, because Health Canada sees this as the component, the active ingredient, they would like to be more specific than broad. Thomas?
Sorry, the question being the low, medium, high feasibility of the label?
Instead of putting the actual number, you just put this is a low category cannabis medium category, and you don't put the label, the percentage.
No, we
Would Health
shouldn't have
Canada
to
even
change the
accept that?
label to being something different because we can't make sure the values being put on are wrong in the first place.
Yeah.
We should go back and make sure that the values being put on are right. That's the obvious solution here. So,
Yeah.
I'm going to point out to certain US jurisdictions, like there was a beautiful paper that came out in Nature looking at Washington State, where they did a statistical analysis of all of the THC potency values reported for dry cannabis flower. And they were able to, it will follow a normal distribution curve. They were able to identify clusters on the high potency side that correlated with certain laboratories and certain producers and go and perform investigations based on that statistical analysis only. No wet work involved. Beautiful. And you could instantly start looking in and identifying cases where you need to do a further investigation. The fix needs to happen before the number goes on the label, not like, oh, well, we can't possibly put a correct value on this label, therefore let's just switch to low, medium, high. In any other industry, if you were falsifying the data that went on a consumer product, if I received a pharmaceutical drug that didn't meet its expected dosage and I just boosted the value to meet it, I could literally go to jail. If we were boosting the protein content of corn so that our customers could get a higher grading for export, CFIA would shut my ass down so quickly, and it would be bad for our export reputation, right? Because our agricultural expertise. Unfortunately, we just get treated like a bunch of stoned 14-year-olds in the school playground where Health Canada doesn't really feel like it's a real industry that they need to regulate. It's like our government pours billions of dollars into subsidies to make electric battery plants, but with this manufacturing industry that's sprung up overnight, here where people are ready, willing, and able to start producing this, there's no action from them to do it. So I genuinely believe that we need, it's like, I don't want to interact with Health Canada any more than I need to, but it to me seems like the only fix is to have a regulatory agency start to take these kind of actual analysis and actually looking at these values to make sure that these consumer goods are being labeled properly. And not all of this goes to just, stoners. Like some of this goes to medical patients as well too, right? It's not fair to have a product that people are using for medical applications, especially be mislabeled for potency.
Thank you.
Thomas, I'm going to ask you a question if you don't mind, because I find it, so I love the passion. I think you're spot on. I think there's enough data out there that if Health Canada really wanted to, they could start looking and identifying a lot of the trends. I'm just stuck on the comment you made earlier where you don't feel that the lab needs to basically speak up. I guess I find that
Sorry, I guess speak up. The question I had interpreted, Nico. Sorry, finish.
You don't mind. Yeah, sorry. I guess what if the lab is seeing something that is questionable, I would feel that the lab should be questioning those to your point, yes, maybe you don't want to speak up right away and report anything, even though there is an anonymous line at Health Canada that allows you to report something. But if a lab is seeing multiple times something that seems to be a little weird, I feel that the lab should be kind of reporting and a little bit more questions.
Sure, I guess if we had very clearly defined frameworks on what they identified as suspicious, and there was a clearly defined regulatory matrix that said if you have a sample that is categorically classified as dried flower and it reports above a certain amount, and all that was written into regulations that all labs were expected to follow that or generate some sort of internal warning when that hit the system, that makes sense. I guess I was responding to Nico's comment more in like the self-regulating aspect. Whereas if you rely on labs to self-regulate that, the labs that are corrupted anyways aren't going to do that, right? So if it was very clearly articulated, then yes, that could be a possible mechanism that would deter at least client side and even, also lab side inflation in the data, because any results that came back would trigger an investigation. So I was answering that in the context of like self-regulation versus like articulated regulation. Again, I'm saying I want the articulated federal regulation. So if that came in, I do agree that would actually be a good thing. And again, I just keep in mind for a high volume laboratory, we don't always know what's an R&D sample, right? So, and I think it's a great proposal. I just always want to point out the complexities. Say clients are doing someone who's doing breeding, when they're doing their pheno screening, they may want to analyze on the dry weight basis because it gives a more standardized number for what they're looking at. So if they're sending in their samples dried, but then we're setting off a red flag because something didn't have the right moisture or something like that, it does introduce some complexities into the workflow. But again, I don't think we should let the fear of these complications stop us from trying to make improvements. So I do correct what I was saying before. I do think from an articulated regulatory standpoint, it would be an effective step. I don't trust the industry to self-regulate into it on its own.
That's great. Thanks, Jonathan, for clarifying that. It's a good two sides of that answer, actually. So thank you, Thomas. Dave, a question from the audience once again. High THC could be achieved with infused cannabis flower, so sprinkling on the flower. Is it easier to change policy to allow for higher purchasing limits, or is it easier to breed a 40% THC plant?
I'm sorry, I'm not sure I understand that question. So could you?
Is it easier? Yeah. So is it easier for us to go to the regulators and say, let us sprinkle the buds and make a higher potency bud, or is it easier to just grow a 40% THC plant?
Obviously, when people sprinkle the bud, when they how does it called? They keef them.
Keefing.
Yeah, when the buds gets keefed, it's a 50-second process. Breeding a very, very high potency genetic obviously takes a lot of time, and I'm not kidding. All of the years I've been doing this, all of the tests that came back, we've barely hit the 30% mark with all the technology and everything that we can do on these plants. And I'm a 40% plant for me, I think is not realistic. I've seen too many cuts, I've seen too many genetics, and it's just and even Nico, and if you look at the results of all the European testing facilities, everything that gets shipped from Canada and get retested, have you ever seen anything over 30%? I mean,
no.
Even with the best growers, even with the best growers. So
correct. Yeah.
Obviously, yeah. There's if you see and now this is something that you're gonna see if labs get better regulations or get better SOPs or methodologies. Everything gets linear in terms of the testing methodologies. What's going to be obvious then is those keefing companies or keefing people that they're tricking their samples, and obviously in the States this is something that's very, very common, and there's a couple of labs that's been shut down because they've either worked with the customers to have higher THC COAs, or just to get more business, they were giving up higher THC COAs, and then in Michigan right now everything that's over 28% get immediately audited. So is this something that can be implemented in Canada? I understand the fact that if you send samples for different testing, if it's super dried, you can reach higher numbers, so it could trigger that 28% for nothing. But obviously, this is something that they've implemented because of this problematic. That it was all over the place, and you were seeing like 40% strains everywhere, and now you're not seeing as many. So there's some ways of
very, very interesting, and I always like to bring examples from the U.S. and it's not because I think, pardon me, that they're doing better than us. It's just that again, their market has matured so much faster, and we can start seeing some trends and hopefully learn from that. So, Jonathan, I know in the intro, I mentioned that you guys are looking at international sales and all that. How does this whole THC lab thing affect that part of your business? Is this something that you must have to take in consideration and align labs? How do you go about all this?
So the time the third party, the international purchaser, will either retest or there's an agreement. Majority of the time they will retest, and then for us, it's not an issue. I think the biggest issue for us is when the buyers come and they're asking for 27, 28% potency and what flowers they're looking for, because that's the trends that they're seeing, right? That's what they think is the norm, basically, especially the new markets, the new European markets that are coming online. I think with social media and bud of the week and things of the sort, we all think that it's normal to see all these high numbers of cannabinoids when finally it isn't the case. So I think once we see Germany come in, we kind of think that things will be tightened up a little bit, and we'll start seeing some people that are going to struggle to get things sold internationally because it's not going to test to the same standards that they are showing on the COA.
A quick comment in the Q&A section. Ethical labs will only need to report when they think a sample is being spiked by producer. The biggest problem is when the lab is the one falsely reporting. So I think Thomas already touched on that, on various aspects on the integrity of that. And really, at this point, it's not the lab's responsibility to be regulators. However, they can definitely show their accreditation and their processes. Do you want to add something quickly to that, Thomas?
Yeah, I'll just point out that on the pesticide side, the way the regulations go right now is that if we pop a positive, we need to report that to the client, and then the client is obliged to report that to Health Canada. So, and initiate an investigation based on that. So again, it's not us doing the reporting. I'm not saying it shouldn't be, but I'm just saying there's already mechanisms in place where certain QC results require you to contact Health Canada and initiate some form of investigation into that lot. So there's no reason why that couldn't be, as long as there was a federal framework in place saying that's what you needed to do, then that would help do that. But for self-regulation, yeah, exactly. I agree. I don't think someone who's already a bad actor is going to compromise their cash flow for that.
And last question, unless a quick one pops up in the Q&A section, but from my side. So, Thomas, how does someone or a facility go about selecting a lab? What should they look for? And, what should they look for when selecting a lab?
Again, part of the challenges that we're facing in industry, like if the answer is how to truly find a lab that doesn't do fraud, that's a challenging proposition because the whole concept of fraud is that you're breaking the rules and concealing it, right? So that's a challenge on its own. In a general, for any QC work you are doing in any industry for manufacturing, some of the things you want to look for when you're choosing a quality lab partner are the accreditations that they carry. So remember, the analytical testing license from Health Canada literally just looks at how safe the labs keep the weed samples. It doesn't actually look at the quality of the science behind the methods at all. So you want to look for things like GMP accreditation, ISO 17025 accreditation. These have shown that third-party agencies have come in and reviewed the quality management system of the laboratory to ensure that it's up to a certain standard. So that's like a starter point to look for. Again, it's not a magic bullet to making sure that bad actors aren't playing. It's just more general strategies for picking a good lab. I would also say something wrong with sending a milled flower sample to a couple different laboratories and then comparing the results that you get in between them. You could even do it with whole bud, just keeping in mind that there is some variation between the buds to begin with. This is another way, like a potency test is not that expensive, but this is a good investment to actually generate like real data comparing different laboratories in between. There are also a person who's really been a big driver for, I would say, kind of tackling this issue has been Rob O'Brien at Supra THC out in BC. Actually, Organogram has been working with him, and Organogram has been sending out samples. We just received ours recently for these kind of voluntary interlaboratory proficiency testing schemes. So Rob, we do this already as part of our ISO 17025 accreditation, except those results aren't published. Rob from Supra out on the West Coast, in conjunction with Organogram, has been organizing these ring testing schemes where we get a blind sample that everyone else gets as well too. We test it and we report back, and that's publicly posted. So You can look at the test results from some of these studies as well too. Will help you kind of like, show who might be a more honest actor compared to those who might not be. Again, there's always good to have an on-site audit as well too. I've heard stories of people going to on-site for some of the bad actor labs who I've been warned I'm not allowed to name here, and interviewing the analysts who are doing the workflow and getting, and asking how much subsample do you use in your workflow, getting strange answers like, oh, it depends on the client. Like not the SOP or something like that. So always an on-site audit is a great way to go and identify any quality concerns or make sure the system is performing pretty well. You can talk to the analysts who are doing the work as well too. So those are some of my strategies I would suggest for starting to choose a quality lab partner.
Awesome. Thank you so much, Thomas. And I just opened up a can of worms and we're getting out of time now. When I mentioned that if anybody else posed a question, I got 3 more questions, but we are out of time. What we'll do, we'll take these questions and we'll put them in our summary of the Canna Talk. And hopefully get some answers for you guys. So I really appreciate the audience participating in this one. This has been very, very good. So this is our time for today. So thank you for the 3 panelists. Your expertise was greatly appreciated. I love the insights you guys brought. As we see, our 10-year-old industry, 5 in the recreational, is not perfect, still needs some refinement. We need to keep communicating, as per Jonathan, keep communicating and addressing these issues. That's a big word. And we will all benefit from this. Thomas, I'm really happy to hear about these cross labs testing. So obviously there is a lot of hype around this conversation and people seem to want to resolve this. So thank you, Canna, once again for organizing Canna Talk events like these. The industry, we need this voice and we need platforms like this so we can communicate and get these out. If you would like to participate on a panel with us or have some feedback, please email cannatalk@canna.ca. We'd love to hear from others, some topics, or some volunteers from some panelists. And remember to subscribe to the Canna newsletter on their website at www.canna.ca//Canna Talk to stay updated on all their events and subscribe to their newsletter and upcoming events. So thank you very much, everybody. Once again, great panel, and look forward to the summary of this. Thank you very much.